We're going to talk about the Huntington's disease family, and I know some of you in the audience, unfortunately, are well acquainted with Huntington's disease, but probably many of you are not. It's not a very common diagnosis. This is my wife, my daughter, my son walking along in the west coast of Iceland some years ago. The humps that you saw about basic science, and then the clinical translation, and then the end point of getting into the clinic, I'm mostly straddling those two second humps, mostly the third. I'm in the clinic. I'm a clinician primarily. I see lots of patients, including a substantial number of people with Huntington's disease, and have for a number of years now. These are some disclosures I wanted to acknowledge, especially HD Care, that many of you here are involved with, which has been very helpful in terms of supporting our efforts both in the clinic and the research side as well. Well, what is Huntington's disease for those of you who have not encountered it previously? It's a neurodegenerative disease, which doesn't really say a lot, but it belongs to a family of other things that you're probably familiar with. Almost everybody knows somebody with Alzheimer's disease. It's so common, and many of us know people with Parkinson's disease, another neurodegenerative disorder. Lou Gehrig's disease is fortunately uncommon, but everybody has heard of it in some fashion. The others, there are several other neurodegenerative diseases that belong to this family, including multiple systems atrophy, cortical basal degeneration, things that probably, I hope, none of you have heard of or ever will encounter these diagnoses. They are unpleasant. This is a slide that often comes up in discussions about Huntington's disease, and it does show the impact in a fairly dramatic fashion on the brain. It's not just a selective injury to certain brain cells, but has a very diffuse impact on the brain itself. So you see on the left side a normal brain and these rather generous, I'm trying to look for here we go. This is the caudate nucleus. These little slits here are actually fluid-filled spaces, ventricles, that we all have and should have in our brains. And then you see what happens in people with Huntington's disease who've had it for a while. Not just really the loss of these sort of bulbous-looking structures, you can barely see them anymore. They're hardly present. But also there's injury or loss of brain cells throughout the brain, causing these fluid-filled spaces to increase in size, expand as brain cells are lost over time. So that is a quick shot of the diffuse impact of a neurodegenerative disease on the brain itself. This is the human impact. I'm going to show you several examples of this. These are people from my practice over the years. Some of you will recognize Christian. I can't remember how old he was when I made this video just down the walkway at the Gottschalk Medical Plaza. I think he was in his 30s. Christian inherited Huntington's disease from his father, and I believe his son acquired it from Christian. I never met his son. His ex-wife wouldn't allow him to come into the clinic to be seen. This is another one of my patients. This is actually just across the walkway at one of the clinical research sites that we utilize. And these are some of the classic manifestations of Huntington's disease. This is not medication-induced. This is disease-induced. These involuntary movements, which are called chorea, not the country, but C-H-O-R-E-A. With your right hand open. Just walk normal. As you know, she says, I say just walk normal because I'm making a video. No, I am walking normal. This is her normal gait. Again, that's the conspicuous feature of Huntington's disease, in when the disease is in sort of fully expressing itself in the motor manifestations. This is a woman I met some years ago. I met her actually when she was 83 years old, and that's how old she is in this video. She's also having chorea. Okay. Can you touch your finger to your nose? Back and forth, again like this. And do it with your left side now. Okay. All right. With your right hand, can I have you open and close like this? With your right hand, can I do this? now with your left hand. Good. And with your hands in your lap, like this. Like this, with your right hand. Maybe tap like that. Tap and then turn it over. Okay, good. All right, that's fine, good. Here she is about 2 years later. How are you feeling? Okay. She is dramatically different. She has lost a huge amount of weight. She still has the choreic movements, but they're lessened, which happens in Huntington's disease as time goes by. The chorea tends to subside. People lose weight. Okay, you can set your hands down now. Good. And then lastly is a woman I saw some years ago. She was about 30 years old in this video. And had onset of symptoms when she was around 18. Shaking is trouble. Can I have you, we did this before, can I have you touch my finger? So as often is the case in young onset or juvenile onset Huntington's disease, she doesn't have the classic choreic movements. She has more of a Parkinsonian presentation. She is stiff. She is slow. She looks like she's got severe or advanced Parkinson's disease in this video. Touch your nose. With your right hand, like you open and close like this. Now with your left hand. Good. With your hands on your lap. You're able to snap like this with your right hand. Now with your left hand. So what is not captured in these videos is that every one of these patients was like you and me when they first started their lives. Unfortunately, this woman had changes very early in her life. The woman that I showed 2 video segments of, it occurred much later in her life, but they all have in common that they started life normally. They had their families. They're somebody's wife or daughter or husband or friend or family member, and then they are affected in a terrible fashion by this neurodegenerative disease. And what's also not evident in these videos is that the cognitive changes that occur with this diagnosis often typically precede the emergence of motor manifestations. So people perhaps for years will have cognitive or behavioral changes that are not clearly recognized as being part of the disease process. They may be attributed to misbehavior or some other personality disorder. For example, the woman that I showed the 2 segments of started having psychiatric symptoms and was diagnosed as having bipolar disorder onset late in life, which would be extremely uncommon. And it was only when she started having the involuntary movements and was referred to see me that the diagnosis became evident. I mentioned, and we'll talk a bit more, that this is an inherited disease. This last young woman and the woman who I showed 2 segments of had no known family history of Huntington's disease. This young woman's father disappeared when she was a child, and probably was the person who transmitted the gene to her. And the older woman that I showed you just before her was adopted. She was born in England and had no contact with her biological family, so didn't know about the family history of Huntington's disease. It is an inherited disorder. It does result in progressive and neurological decline that's invariably fatal. People don't survive with Huntington's disease. They ultimately do develop complications that are fatal. It's inherited from either parent. It's been known for quite some time where the gene resides, on which chromosome, and also in 1993 the specific gene was identified by a study that Leslie actually was involved with, going down to Venezuela and looking at family members, many family members with Huntington's disease. There's a large group of people in Venezuela with Huntington's disease, and by doing something called linkage analysis, the gene was identified in 1993. So we know that we've known the chromosome for a long time, where the gene resides, and the specific gene was identified. And exactly how that gene is leading to what we just saw in those videos is not yet clearly elucidated. Every child of a parent with Huntington's disease has got a 50-50 chance of also getting that gene, and at some point getting the symptoms of the disease. The average age of onset is around 35 to 40, but as we saw, there's this young woman who was 18 when she first started having symptoms that ultimately were recognized as being Huntington's disease, and that other woman came to my attention when she was 83. The average duration of the illness is about 15 to 20 years, with death resulting from infection, complications, aspiration pneumonia. People become debilitated, bedbound oftentimes, and are prone to infections with chronic illness, including aspiration pneumonia and ultimately death. Cardiac causes actually are a leading cause of demise in people with Huntington's disease. And sadly, particularly earlier in the illness course, there's an increased risk of suicide. People have often seen their parent, for example, decline, or one of their parents decline with Huntington's disease over years, and they say, I don't want that fate. And when they're able to act earlier in the disease course, people have a higher risk of taking their own life. It's been published that people with Huntington's disease have about a 6-fold increase compared to the general population of the same age for taking their own life. The diagnosis remains based on the motor manifestations and recognizing those motor manifestations for what they are, which in these videos it was largely obvious, but they can be subtle and sometimes difficult to recognize. And so if the family history is not evident, the disease can often be overlooked or attributed to something else. As I mentioned, the cognitive changes typically do occur earlier than the motor manifestations do. Since the 93 identification of the gene, we've had a blood test available to confirm the diagnosis. So if somebody shows up in my clinic or a doctor's office, we can do a blood test. If they are concerned that they may have Huntington's disease or that their parent had Huntington's disease, we can do a blood test for the patient. It's quite simple. It's a little bit more complicated in terms of identifying the gene. It takes some time, a couple of weeks or more to come back, but we are able to identify people who are carriers of this gene. What we're not able to do is anything about it. We can find people who are at risk, or I had a patient recently who came to see me. She's in her 30s and her mother had Huntington's disease. She arrived with her father and she wanted to be tested. She said she'd been thinking about it for a long time, and she was single, was not planning on getting married or have children, but she did want to be tested. And we talked about it a lot, about whether it was helpful or not, because what do you do with that information? As long as you don't have the result of being positive, you can still say, well, there's a chance I don't have it. I can live with a sense of optimism about my future, that perhaps it will come, I don't carry that gene that caused the death of my parent. We talked about that at some length. There are other implications. Insurance, life insurance, long-term disability insurance. You can't get that once that gene has been identified. And we had a lengthy conversation about it, and she ultimately decided that she wanted to be tested, and sadly, it came back positive for her. We know that the gene is abnormally expanded, and Leslie can talk about this more. We know what the normal size of this gene. There are certain repeats in this gene that become larger in Huntington's disease. We know that people who have 40 or more repeats in this gene will get Huntington's disease. They will have manifestations of the illness at some point in their life. We just can't say when, or exactly how it will show up, or how it will behave over the course of their life, other than it will likely cause both motor and cognitive decline and ultimately demise. Now, these other areas, this reduced penetrance, meaning that, well, if you have got 36 repeats, now maybe you won't get the disease or it won't show up, but it's less clear. That's a very tricky situation for patients to be in when they've got this sort of gray zone and not really clear about whether they're going to have the disease or not. When the gene was identified and the test became available, most of us thought, well, we're going to have a lot of people requesting to be tested before any symptoms show up. And that, in fact, has not been the case. People oftentimes decided they simply didn't want the information, and I can understand that quite well. The implications of the gene size are not only about whether somebody gets the manifestations of Huntington's disease or not, but also roughly it correlates to age of onset. So the bigger the gene expansion, the earlier the onset. So the young woman I showed you with Huntington's disease from age 18, I don't remember her repeat length, but I would assume it was quite large. The woman who was tested recently, who has no symptoms, unfortunately has repeats. I believe hers were 44. So she's well into the range where at some point she will unfortunately develop symptoms of Huntington's disease. The size of the gene may correlate to the rate of change, but that's less clear. And there's no real correlation to the size of the repeat expansion to the way it shows up in terms of psychiatric type symptoms. So for an individual, I can't tell if somebody like the woman I saw not too long ago who came back with her positive test, I'm not able to tell her at what age she individually will start showing symptoms. Nothing really predicts the severity of the illness. Leslie and I both know somebody we've known for years who has symptoms of Huntington's disease and she's in her 70s now. I still see her on a regular basis, about every 4 to 6 months. She's been in some of our clinical trials, and she's doing quite well despite her carrying this abnormal gene and having had symptoms of Huntington's disease for some time now. And nothing really predicts the rate of change. We can't tell people on an individual basis how you will be after symptoms start in 5 years or 10 years. You can fall back on groups of patients, but it doesn't really help on an individual patient. What I can tell some of my patients, because they have seen their parent oftentimes suffering with this diagnosis, is that their parents' experience with Huntington's disease does not necessarily reflect the parents' offspring, how they will be experiencing Huntington's disease. This is a slide that's often used in papers or review articles about Huntington's disease. It kind of irks me a little bit, but it does capture, in a sense, the essence of Huntington's disease with motor manifestations, cognitive issues that I mentioned, and psychiatric symptoms. I sort of personally link the psychiatric and the cognitive things together. But this actually describes most of the people I see in my clinic, including people with Parkinson's disease who have motor manifestations. They usually have cognitive issues, and they do sometimes have psychiatric issues like depression. They have hallucinations and delusions, not uncommonly in Parkinson's disease. So this is a common characteristic, I think, of neurodegenerative diseases, this mixture of both motor and cognitive changes. This is a word I can never pronounce, and I challenge you to say it quickly. But this is something else that this often comes up in discussions about Huntington's disease, this unawareness that patients sometimes exhibit, or oftentimes actually usually do exhibit, in terms of their own manifestations. But this is also true of my Parkinson's patients. They often don't recognize the severity of their impairment, the difficulty that they're having, both with motor manifestations, also cognitive issues, and the impact it can have around others in their family, for example. This is Huntington's disease in particular is like a bomb going off in a family, and those of you who have been close to somebody with Huntington's disease know that quite well. One of the more difficult conversations I have with my patients, not just with Huntington's disease, but also with Parkinson's disease, is their driving. They often have poor insight about their driving safety, and this has led to some pretty serious discussions, difficult discussions for me and my patients. And also the impact on those around them is often unrecognized by patients, how this is having an impact on others. There are very few treatments. This is, I went into medicine because I want to make people better. I want to be helpful to people and improve the quality of people's lives. I hope I do that with my patients, including those with Huntington's disease. But the tools we have are pretty limited right now. There are a couple of medications that have been around for a while to try to reduce those involuntary movements, and they're helpful. And that can make a big impact. The first fellow I showed you, Christian, who is sadly deceased and has been for some time, but he was still employed when I first met him. He was working in maintenance, and walking down the street, he was stopped by the police because of those movements. They thought he was on drugs, and they pulled him aside. So those involuntary movements can be attention-getting in ways that people don't want. I do treat some of my patients for their psychiatric symptoms. People are sometimes aggressive or hostile or angry with family members, or they may even have delusional thoughts, and that can be dealt with by medications. The cognitive issues that we've attempted to treat with a variety of trials, medications that were approved for Alzheimer's disease, for example, but they have not been successful so far. And those can be very troubling as well. And we offer people rehabilitative therapies like physical therapy, occupational therapy, and speech therapy to address speech difficulty and swallowing problems. But there's no treatment presently for the disease itself. We would like that a lot, as all of our patients and their family members would as well. This is just so frustrating to have so few tools and have the disease itself. You know what the gene is, you know where it's coming from, and not being able to fix that particular problem. Parkinson's, if you've encountered Parkinson's disease, you know it's not just tremor, stiffness, and slowness. There's a whole long list. This is a partial list of things that torment people with Parkinson's disease. Interesting to me is that this same list has really not been generated in Huntington's disease. I think it's probably the case. This is not just a brain localized disease. I suspect this is a systemic disease. And the more we know about it, the better able we will be to treat it, to recognize it earlier, develop therapies that can modify the disease course. There is evidence of this disease outside of the brain. One of the highest concentrations of the abnormal protein that's coded by this abnormal gene is in the testes in men. But as I mentioned, cardiac deaths are actually fairly common with Huntington's disease, so there's evidence of it involving the cardiac muscle, other muscles, skeletal muscles, not just the heart. Neurodegenerative diseases have some common features, and those of you who work in the field know this quite well. They're very selective loss of certain brain cells or cells in the spinal cord. The energy-generating little organelles inside the cell, the mitochondria, are commonly affected in all neurodegenerative diseases. There's something called oxidative stress, which is a chemical process that can cause injury to other components of the cell itself. And there's often, or typically in these neurodegenerative diseases, an accumulation of proteins. In Parkinson's disease, it's something called alpha-synuclein, and in Huntington's disease, it's Huntington. And sometimes these proteins are sequestered inside the cell, and other times outside of the cell, such as in Alzheimer's disease. There are common clinical features, as I mentioned, both cognitive and motor and other manifestations as well. And because of the commonality of some of the pathology, the biochemical processes, the clinical manifestations, I think it's reasonable to believe that things that we develop for Huntington's disease, which is not common, will be applicable to these other neurodegenerative diseases, some of which I've listed. These things. So what we can do for people with Huntington's disease, I think will shed some light on what we can do for people with Alzheimer's disease and Parkinson's and Lou Gehrig's and so forth. This is my last video. Going back to windy Iceland by a waterfall. We all want what is best for our children and for our family members, and this has been a very tough nut to crack, to say the least, a very hard thing to see in my practice. Without having the tools that we need to really intervene in the disease process. I'm going to stop there. We're going to pause for a moment while we switch computers. That's Iceland again. And if there are any questions or comments, I can take a little bit while we're changing over. Thank you. You're welcome. I'm always a little bit reluctant to show these videos any longer about patients because they don't capture the person. They capture a segment in time when people are having manifestations of their disease. And I don't think it's fair. I don't think it's a fair representation of their life, their connection to their families, or the impact that the disease is having on not just on them in so many ways, but also on their family members as well. But it is a quick way, I think, to make an impact of how horrible this disease truly is. Yeah, the question is, are we involved in any clinical trials? We've been involved here in clinical trials in Huntington's disease for quite some time. We have a couple going on now. One is just a information gathering study called Enroll HD, just so we could learn more about Huntington's disease. And that's been, it's an international study with multiple sites around the world. And we are one site for that. We are doing a small pilot study of a medication called fenofibrate, which also known as Tricor, which is a lipid-lowering medication because there are reasons to believe it may have a positive impact on the actual disease process. I saw somebody earlier today who is involved in that study. I saw a patient in my clinic yesterday who was a participant in that study. We are, and Leslie can talk about the stem cell project, we're also involved in a study collecting information about collecting spinal fluid from patients with Huntington's disease. This is based out of London with also multiple sites around the world to try to better characterize chemical changes that we can measure in spinal fluid in people with Huntington's disease. I just received notification by email early this morning that we are selected as a site for that study.
Cool. For clarity?
Yes.
Okay.
Oh, Francis. Can you talk a little bit about oxidative stress and how that affects people with Huntington's?
Yes, Leslie can. She'll do a better job.
We don't totally know. It seems to create certain species in the cell that are not helpful, that are deleterious. Actually, the fenofibrate drug that Neal's talking about is to help cope with that. It also copes with mitochondrial problems and helps maintain energy balance and things. So we don't totally know yet what oxidative stress does per se. It can do many things to a cell, but we don't know exactly how it's working in Huntington's. So what I'm going to do is then build on what Dr. Hermanowicz was talking about from the clinical standpoint and some of the work we're doing in basic science specifically relating to stem cells. We have a number of projects going on actually using the CSF that Ed Wild's been collecting. Student in the lab's been using that, so I'm really excited. We're an HD Clarity site now. And many of the other features of the disease, but I'm going to focus on the stem cell part. As Neal was talking about, Huntington's disease caused by a mutation, a single gene mutation in the Huntington gene. And it's caused by a repeating CAG repeat unit. It's at the very tip of this chromosome. And when this expands beyond the normal repeat length that he was talking about into the disease range, which, as he was mentioning, between 40 and above is the disease range, it changes the structure of the protein that's made. So the DNA encodes this CAG triplet repeat, and that's ultimately made into a protein encoding what's called a glutamine. It's a polyglutamine repeat, many glutamines in a row, that causes the disease. And this is a very dominant mutation, so when you have this in the cell, you start to begin to have all these deficits occur within a cell, in particular in neurons and other brain cells. So it's really a genetically programmed degeneration of nerve cells and other brain cells. And as you saw, it has a profound impact on the brain, on the whole brain, as you heard. And what we strive to do is really look at the simplest form of this in a cell, what is going on with that mutation present in a brain cell. And so the way I divided the talk is I'll talk a little bit about the IPS work, induced pluripotent stem cell work that we've been doing, using an example from the HD research, and also sort of where we're going with that, using an example from ALS, and then this work on stem cell transplantation for HD. And we've had a number of models ever since 1993 when the disease gene was cloned. There have been tremendous number of worm models, fly models, mouse models, yeast, every kind of model you can think of to mimic the disease aspect, put the human gene into a system and mimic the disease. But we really want to look at human cells. And so this has been a very powerful approach to use stem cell biology that you heard from Dr. Anderson, where there's multiple types, adult stem cells, embryonic stem cells, and I'm going to focus on the induced pluripotent stem cells. And this is really where we're reverse engineering a mature adult cell, such as a skin cell. You can take a skin biopsy. Several of the family members here, we've had that done. And you take that and go back to its embryonic state. So in the developing embryo, when a baby is developing, what happens is that those cells form mature cells, and you heard about this from Dr. Anderson. But if we reprogram them with specific factors, this can be almost any cell, in this case a skin cell, it goes back to that pluripotent cell that can go then into any cell type we want to study. So this has just really transformed our field and every field to study these neurodegenerative diseases, because we can now look at a neuron in a dish, because we could never look at a human brain cell before. And just show thanks you to the HD families who have donated those skin samples. This is Frances who's here. Frances's daughter Marie was our first person that donated her cells in the clinic. And what I was going to do is show an example from HD. As you heard from Dr. Hermanowicz, we've been studying brain cells, but it affects many different types of cells. So within the brain, the blood-brain barrier was thought for many years to maybe be impacted, maybe not. This is the barrier in the brain that preserves the integrity, doesn't let different things that shouldn't go into the brain across and get into the brain. And just for a second here, can you hear it? Yeah.
The blood-brain barrier, such as other blood barriers, is a remarkable anatomical adaptation. Its most striking structural feature consists in endothelial cells, tightly joined by tight and gap junctions.
We'll stop there. Basically, these are specific types of cells that form this barrier in the brain. And we wanted to ask the question, are these affected by the presence of the mutation? I should mention the HD mutation is everywhere in the body. Every single cell in the body has that gene and now has that mutation and carries that. So we differentiated. This is a project done by Ryan Lim, who's now a project scientist in the lab, where we took those IPS lines from patients, guided them into these brain endothelial cells that you saw the structures of in that video. And they remarkably look like a blood-brain barrier. They form the structures that have barrier properties. And we used what we call systems biology. So we're trying to look at what are the earliest changes we can find. What are the things that dictate what's going wrong with that particular cell? And we've done this for neurons. We've been doing this for a number of different cell types. This is just an example from the endothelial cells. Again, this is work done by Ryan. Here's the blood group. And so what we looked at first was the gene expression changes. So in the cell, you have DNA that encodes RNA that ultimately encodes the proteins that make up that given cell type. They carry out the functions of the cell. And so we looked at the transcription, so the expression of the genes in that given cell to see what was made appropriately and what might be made inappropriately. And this is just a very complicated figure here, but from that knowledge of the gene expression, you can start to put these genes into what are called networks. You can start to build a picture of what's going on in that cell and start to understand what might be going wrong. And one of the things that came up here is that a particular pathway that's composed of a number of different genes that have a specific function seemed to be impaired, and what that caused was an increase in what's called angiogenesis. So what does that mean? So for instance, this is then tested in a human brain. You can see that in the HD brain, there's the presence of that band. Doesn't matter, this is a Western blot, but you can see clearly here you have a very significant amount, and in a control brain you don't. And that came out of what would be predicted from this kind of network-based approach. And ultimately what it meant is that this process of forming blood vessels in the brain that's part of the blood-brain barrier was impaired, and you had actually too many of them formed. Too much of this angiogenesis, the production of blood vessels that could occur based on these cells, the information from these cells. And so what we can do is we can study these processes then and figure out ways to intervene and restore normal function to the cell. So that was what was done in this particular study. And what we found was that then we had a model for the blood-brain barrier in a dish that we could study. It demonstrated that that might be contributing to some of the features in Huntington's disease, and it could potentially inform some of the therapies we might take forward. And this really is based on the ability to look sort of genome-wide in these human-derived stem cells that you can differentiate. And what we've been learning, I think, over the years, and in some ways Neal was alluding to this, is that many of the drugs that we've tried over the years have failed in clinical trials because of lack of efficacy. And it suggests that we just don't know the right thing to target. We don't know what we should be treating for those diseases. And this is just work that's sort of an offshoot of the Huntington's work in ALS, or motor neuron disease. And let's see here with Stephen Hawking's, the Ice Bucket Challenge, who's been funding a lot of this work. And what we know, for instance, in ALS, is that there have been a lot of drugs tested that have been tested too in the disease to try to change the course of the disease. Only 2 of these have had any effect, and it's been very, very minimal. Again, not really understanding what we should be targeting to try to treat the disease. And so this applies to all of these diseases. Is Alzheimer's disease, is Parkinson's, is Huntington's, is it one disease just lumped together, or is it a lot of different subsets? So do patients have specific aspects to them that dictate how their course of disease is going to progress? And that's, as you heard, it's even the case in something with a single gene defect like Huntington's disease, where you have different manifestations, different ages of onset, subgroups of patients that might respond differently to a particular drug. So we're using these kinds of approaches, going from the human patient, iPS cells, doing these kinds of large genomic studies, using what's called precision medicine, so getting the right drug for the right patient, and taking that back to the human patient in clinical trials. And this is, we're part of a large network called Answer ALS right now that's taking 1,000 patients with ALS, sporadic ALS. This is mostly a sporadic disease without a clear genetic cause, and turning those into iPS cells, doing this kind of big data approaches, and looking at the networks that come out, like I showed you a minute ago for the blood-brain barrier. And this is really based on this type of idea. And you can do this so well with precision with iPS cells.
Network models of disease to enable us to translate this wealth of complex biomedical data into better diagnosis and treatment of individual patients. How do network models help us to better understand disease? Network models enable discovery of key mechanisms, such as specific genes, transform a disease network back towards a health state. For example, if we amplify this node, the network is mostly stable. If we suppress this node, only a few neighboring nodes may be affected. However, if we modulate this node, large swaths of the network are affected. This node is a key driver and is important because it identifies a specific target in the network that can have a major impact on the disease.
So this is what we're trying to accomplish when we're doing this type of work. As I showed you with that Wnt pathway in the blood-brain barrier, that was a key node. That was a key regulator of some of the deficits that were occurring. So we're trying to understand what this might be in these various disease subtypes, and using these iPS cells really have powered the ability to do that kind of work. So we're kind of at its infancy, but we're using this approach in Huntington's disease, ALS, on campus here at UCI within the stem cell building, iPS cells from Alzheimer's patients as well, and using these kinds of approaches to get at what's going wrong. So the second part of this, and finish up with this, is regenerative medicine for HD, using stem cells as a way to treat the disease itself. And as you've heard about, neurodegeneration is really this progressive loss of structure in the brain, including the death of neurons. And what this communication between neurons in the brain, for instance, these electrical signals that connect, that trigger the communication between cells, is really lost in Huntington's disease and many of these neurodegenerative diseases. So you have an inability to connect. What we're hoping by transplanting stem cell products is that we can prevent that degeneration. We can not only sort of nurse these cells, we can provide some support for the existing cells so they don't degenerate as quickly, but what we're finding also is that we may actually be able to restore some of these circuits in the brain that were lost using stem cells. This is just the experimental design of the preclinical studies we've been doing, and this has again been funded by CIRM all the way along, from discovery grants to a preclinical grant, and now we're hoping to go into clinical trials in the next about 2 years with this. But the design is that we take a mouse model of Huntington's disease. These are human neural stem cells, so these are stem cells that have been started along the path to a neuronal lineage, to neurons. But they're still in their very immature state. They can become an astrocyte, a neuron, an oligodendrocyte, different types of brain cells. And we have a human line that we've worked on for many years with UC Davis as well. That is GMP grade, so it's good manufacturing practice. It can go into a human individual. And we implant those cells into the striatum, the region that Dr. Hermanowicz showed you degenerates in the disease. We test behavior, collect tissue, and look at it and analyze the outcome. You can see sort of this little strip of human cells that are transplanted in this particular mouse here. What we find, first thing that we had to do was a lot of tests of how to get the cells to survive. They do survive. This is just an example of a stain of a brain tissue showing these red dots, which are human cells that are co-staining with a neuronal marker, a marker of sort of early guiding down a neuronal path. They're neuronal precursors still. So they do survive and they start to become neurons. And this is a very rapidly progressing mouse model, so they're only in there for about 4 to 5 weeks, very, very short time period, but they start to go along this path. They help the mice in a number of behavioral tasks. One of these is what's called a rotorod task. And these are the mice just running on a little treadmill. Wild-type mice can do this for a very long period of time. HD mice start to lose the ability to hang on. And that progressively gets worse as the mice become more diseased. We use this pole test here also, where you pick it up by the tail, it just runs right down a little pole. HD mice are very impaired in their ability to do that, or to grip onto a wire mesh, which is actually very analogous to a test that's done in human patients as well. And there's a certain percentage of rescue. This is, again, a very aggressive model of the disease, but you do see improvement in those behaviors. In a longer term mouse, so this is a mouse that lives for 2 years, and has a much longer course of disease, you also see these having a drastic effect on this ability to stay on a running wheel. This down here is the HD mice. These are wild-type mice, and these are the mice that have been transplanted with the human neural stem cells. So they have a absolutely significant rescue. This is where the circuitry comes in. Now this looks like a complicated slide, but what I want to show you, this is what's called an electron micrograft. So this is looking at the tissue at a very, very deep level from the mice that had been transplanted. And first of all, what you see here is this cell. This is a human stem cell here, differentiated human stem cell. They differentiate into really nice neurons because they're in there for 8 months period. Those are human stem cells, and they have connections to other human stem cells, and they also have connections to the mouse tissue. So from these kinds of experiments, as well as things that measure the physiology in the brain, we know that there's active connections being formed with the human stem cells that are transplanted. So it's really been a very, very promising project. We're very hopeful for this. Our next steps are to go to what's called IND enabling activities. We've had our first meeting with the FDA to discuss this project and where it should go next and what types of safety studies we would do. So the next step are to actually do these very involved safety studies in mice and get all the sort of really make sure that this is not going to be harmful to anybody and that it will have hopefully some efficacy. And all of these things are, take a lot of people. These are large groups of individuals. This particular work is done in collaboration with UCLA, with Mike Levine, with Gerhard Bauer at UC Davis, Charles Meschel at OHSU, a number of clinical advisors who have been helping us with this project, as well as, of course, Neal and Ed Minucci, Steve Kramer, and really spearheaded by Jack Riedling in the lab. This is the lab at a recent gathering on this beautiful 4th floor that we got to move into recently. And it's the work with the stem cell, stem cells are just probably the most exciting thing that I've been doing since we were looking for the gene and got to use all those new techniques to do the human genetics work early on. So I'm quite excited about it and very hopeful. So thank you very much. Hi, Rick.
Leslie, this is really fascinating. And one of the most fascinating things that I find in here is that while these genes are everywhere, they seem to mostly affect these medium spiny neurons.
Right.
So something about those cells, and I think that your iPSC cells in a dish could be really helpful there. So are those medium spiny cells, are they just really poor at taking out the garbage, and could this be a lysosomal storage disease, whereas they don't have active lysosomal enzymes where other cells do?
So yeah, that's a great question. I know I'm going to repeat.
Okay.
Yes. So I will. So basically the question is, what's unique about the medium spiny neurons, and could protein clearance pathways be involved in that selectivity, right? So there's clearly a selective vulnerability for medium spiny neurons. That may be at the level of the energetics that's involved, the whole glutamatergic, the excitotoxicity that's known to occur that fits with the mitochondria. They are also extremely vulnerable to the activity of protein clearance pathways. So if Joan Steffen were here right now, she would say it's all about autophagy, and so, which is her favorite thing to say. So what she has found is, and we know a lot about the normal Huntington protein as well. Normal Huntington protein is involved in autophagy, which is one of these clearance pathways. That is impaired. If you have a defect in that protein clearance pathway, just in general, if you mutate one of those genes, you get a striatal neurodegenerative phenotype. So we know that the striatum is selectively vulnerable to that kind of effect, that kind of impact, right? What we don't know yet is that the whole picture in the medium spiny neurons, or are there other things that are going on? There's evidence for the fact that you don't have correct maturation. So from the IPS work, we know that there are issues with striatal maturation, the ability of striatal neurons during periods of development, but also during adult neurogenesis. So when new neurons are made, they don't tend to mature all the way into medium spiny neurons the way they should. There's structural differences that happen. So they can cope for many years, and then they may be more vulnerable to effects of aging. One of the effects of aging is decreased protein clearance pathways, right? Along with a lot of other energetic deficits, all sorts of other things that happen. But for whatever reason, and that's an active area of interest, and that's one of the things you can do with these iPS cells is we differentiate to medium spiny neurons. And that's something I didn't even talk about. We have a whole program on that. And trying to understand what goes wrong in those cells. What are the very top, pathways that are impacted that are most proximal, right, to the disease? What's the earliest changes we can find?
Yeah, I'd be interested in a comparison of the stem cell that are differentiated into the medium spinous one and cells that have differentiated otherwise.
So this field is so cool because it's very similar to what happened with identification of the disease gene. There was a large consortium that was developed around IPS technology for Huntington's disease. The first thing we did was the HD-IPS consortium was to differentiate into neurons that were enriched for the medium spiny neuron characteristics. And that we didn't even know if we'd see a phenotype because usually to model this disease, you have to have just a piece of Huntington that has the mutation that has a very dominant toxic effect on a cell. And this is in this full-length context in the human, cell that takes a person 40 years to get sick, right? So we didn't know if we'd even see anything, and we did. We saw deficits in bioenergetics, we saw deficits in transcription, deficits in protein trafficking and aggregation or protein accumulation. A number of different features that are characteristic of the disease are in those medium spiny neurons from HD patients compared to controls. And then we've been getting at the systems level that I showed to figure out what are the key mechanisms involved. Yeah, Sid.
First of all, thank you to both of you for helping us understand this. Maybe I missed it, but what is the normal protein?
You didn't miss it. I didn't talk about it. But it's a short time frame. For years we said it's of unknown function. So when it was identified, there was no homology to anything in the genome. We thought we'd know what it did. But it's embryonic lethal if you knock out both copies. So you absolutely have to have it. What we know about it now is it seems to be a scaffold. It's a huge 350 kilodalton protein. It's very, very large. It has domains that match other types of proteins, other yeast proteins, for instance, such as in autophagy. But it seems to be involved in microtubule transport. It binds to microtubules. It scaffolds other proteins. It interacts with 100 different cellular proteins. It's involved in neuronal transcription. It's actually required for neuronal transcription, for production of BDNF, for maintaining normal neuronal transcription from repressor complexes, for instance. It regulates those. So it has a number of different functions, and it's involved in autophagy. Does that clarify?
Yeah, I'm just wondering if you couldn't flood the system with a normal protein, but protein that size is going to be hard to do.
Yeah. And we do know that the repeat impairs the normal function. So we do know that. But what we also know is that's not sufficient to cause the disease. This is a dominant disease that's also impacted by loss of normal function. So, yeah, George?
So when you do these large network analyses to see what is altered when you have the mutation, the temptation is to say, well, this is different, this must be bad. Right?
Oh.
But it could very well be a compensatory, some sort of positive response by the cell, right?
Yep. So that's a great question. So the question is that when you're looking at these networks, how do you know what's contributing to disease and what's helping disease is actually turned on because or off because it's actually helping the disease, it's compensatory. And that's been a big issue in this whole kind of systems biology. And so one of the ways, we've just done this for ALS, we're actually just about to submit this paper any day, is to use a fly model. So there's a fly model of this particular mutation that causes a genetic form of ALS. And so what's been done is to take this network of actually in this case is gene expression, epigenetics, proteomics, everything. Take those systems and then modulate the key nodes in the fly model So you reduce its expression in the fly, and you see if it makes the disease worse or better in the fly. And then you can use these kinds of network-based approaches to see, would that be predicted then to be causal or compensatory? And so that's one way we're starting to get at that type of, and so we're trying to think of ways that you can use, go back and forth between different models and get at those kinds of questions. But we're also going to then modulate those nodes in the iPS cells themselves, right? So, yes, Brian?
What did the FDA say?
The FDA so far, well, so we had our pre-IND meeting, and they were very positive about it. They had things they wanted to see us do in our animal studies. So for instance, don't go 8 months, you have to, if you're gonna use, so we're gonna do our tumorigenicity studies in an HD animal. And that was something we had to discuss with them instead of a nod skid, and they said, well, as long as you go 9 months, that'll be fine. And so that's, we're gonna do that. Things like that, and some of the things they did about cell manufacturing, being able to expand the cells. Those were, they were sort of just very detailed items, but nothing negative about the big picture.
Hasn't biotime cells already been used?
Not that I know of. Actually, Mike West is coming down here Saturday to talk about this some more. But no, not for neurodegeneration at least. I'm not sure about other indications.
Then a follow-up to that
Yeah.
is, are you going to repeat the mistakes of the Alzheimer's field?
Of course.
Treat the worst cases rather than the early.
No, so that's an excellent question. Are we going to make the same mistakes? So we are going to be treating, actually Neal helped a lot with this, our CLIN one we submitted that we're supposed to find out about in about a week if we got. But to use early age.
Sorry, what's a CLIN?
Oh, sorry. This is
started it.
I did. I'm sorry. So CIRM has a number of different types of grants. We had funded for some of the preclinical work. This is the CLIN one, which is the leading up to a clinical trial. And so you apply for this for the funding to do the safety tumorigenicity studies that you've agreed to with the FDA. And you have an 18-month window to get everything ready and file an IND have a productive IND meeting, file an IND with the FDA. So we applied for that.
Population?
Early HD. Yes, that was very specific. We even have exactly the scores, the range of scores for them, and it's going to be early HD, which is what I think people have learned from the Alzheimer's trials. Most of the HD studies, the Ionis trial that just finished phase 2 was a safety trial for an antisense oligonucleotide. Yep, George, and then Francis. Okay, Francis.
So then the clinical trials, will the patient find out if they have it, if they know it's positive, or can they not?
This will be symptomatic HD. Yeah, it's early HD, so but they will be symptomatic HD because there is risk, right? And so it'll be early HD. George?
Yeah, so I realize this is a stem cell institute, but my question is about gene therapy, because one would have thought, this was the first neurological disease where we actually knew the gene. You would have thought that instead of trying to fix it with all these, putting other things in, that we actually try to fix the cells themselves. So
Yes.
I haven't heard anything about stem cell therapy for a long time about Huntington's disease. Is that just dying out, or is it not?
Stem cell therapy?
Yeah, stem cell therapy.
You mean gene therapy? Yeah. No, absolutely not. So it did die down for a little while, but there's probably 5 studies on the horizon right now for gene therapy for Huntington's disease. But what I mean by gene therapy for Huntington's disease is to reduce the levels of Huntington. So to reduce the formation of the toxic protein. The problem is, so they've had a very successful, first of all, there was an antisense oligonucleotide developed by Ionis, used to be called ISIS Pharmaceuticals, it's now called Ionis Pharmaceuticals, but they've developed a way to target total Huntington. So mutant Huntington, you have one copy of normal Huntington, one copy of mutant Huntington. It targets both. Okay, and so you're reducing the levels of of total Huntington in the cell. The problem is we don't know how much Huntington you need. We know if you go too far, it's lethal. We know that it's really important for the brain. We also know that if you have too much, it's of mutant Huntington, it's bad. So there's this sweet spot potentially. And so far they've gone through phase 2 of this trial, and it's been safe in, a year? 6 months? 6 months, with monthly injections of this ASO, and it had target engagement. So in the CSF, you saw a reduction of Huntington, mutant Huntington. And so there's also now approaches there's a phase 3 trial starting at the end of this year. There's approaches to use differences in the mutant gene to target those, which will fit for, say, 70% of the population of HD. So there's ways to try to target just the mutant protein, although that's very difficult. And then delivery is a big issue and all these other things. But it's very much an active area. Plus, some of the targets that we'll be finding, we being global we, will be, there'll be gene therapy around that. So there's a number of vectors being developed for that. There are ways to deliver these things, and there's just huge areas in the HD field and other fields to do that.
Presumably with stem cells, you only have to deliver once.
That's the key. You go in once and it stays there, and it secretes all these wonderful factors that help bathe the brain in helpful things. So anyway, yes?
Well, I was going to ask the question, but thank you for asking it first, because now objects flew in the room. So I'll add to that CRISPR technology.
Yeah. Yes, so...
You can cut the cheese up, replace it, and you're done.
Yeah. So if it were that easy, right? Yeah, that's next week's seminar. But CRISPR technology is definitely an area of interest and under development for Huntington's disease, because yes, if you could target the mutant gene, and either repair it, prevent it from being made, whatever, that would be a great thing. Again, it's delivery, and there are probably at least 3 or 4 different efforts I'm aware of to do that. And it has shown promise in both mice and in non-human primates. They've done some studies in that way, and it has shown promise. There's issues with CRISPR delivery, and there's issues with what are called off-target effects, because they sometimes will target other genes it shouldn't be targeting. So there's still some research that needs to be done in that area as well. Yeah.
How did you guys land on 100,000 cells per injection?
Pardon me?
How did you land on 100,000 cells?
That was the good lesson we got from the Alzheimer's disease field. So we picked 100,000 cells per hemisphere based on work that Matthew Blewton-Jones and Frank Lafrilla at UCI did in a Alzheimer's disease mouse model where they saw efficacy in cognition. So we started with that, and it was very efficacious. So we've been building on that, but part of our studies ongoing right now is to do dosing. So we've done a range of doses, sort of are narrowing it down, and now we're testing within that dose range right now. So we're looking at that. And then we've extrapolated in our application from that number of cells to what it would take to go into a human striatum or a non-human primate striatum, for instance. Yeah. Okay. David?
The human neuron stem cells that you, how are they delivered to the mouse brain?
They're just injected. Injected, yeah, they're stereotactically injected, so we have an apparatus that keeps the head still, and then you inject into a particular part of the brain with specific coordinates, and that's how the human trials would be done as well. It would be in a neurosurgical suite there's a specific surgical instrument that would be used, that is used for injection, but in that case you would be injecting it into multiple sites. So, any other questions for Dr. Hermanowicz?
I have a question for Dr. Hermanowicz.
There you go.
We talked about the symptoms of Huntington's disease, but most of us know that people who have Huntington's disease, they have other symptoms besides the movement disorder. How much in advance of those those movement, symptoms. I mean, how far before that, do the other symptoms, whether it's cognitive or behavioral, I mean, how many years or how much time? Is there any knowledge about, when those start to manifest, or does it not have anything to do with the number of repeats, or is there any knowledge about that?
Yeah, it has been looked at, and there have been a couple of studies looking specifically at cognitive issues in people with Huntington's disease, as I mentioned, a year or more, where there have been evident changes in terms of cognitive parameters that have been studied. But what we don't know is that one slide I showed about other things that can go on. We know about that in Parkinson's disease. We know that there's gut dysfunction, impaired sense of smell, sleep behaviors that change. Years, a decade or more in Parkinson's disease. And that's really been the focus. We've done several trials, and I've participated in some of these, where so-called neuroprotective or disease-modifying therapies have all failed. But one of the reasons that people claim that, or speculate that they have failed, is that we intervene too late. That the disease is already well underway in the brain by the time people show up with tremor and stiffness and slowness. And that may be similar in Huntington's disease. If there are other so-called non-motor, the diagnosis is still, of Huntington's, as I mentioned, is still based on motor manifestations, which we know in Parkinson's disease is probably a late manifestation of the disease. And that may be something similar going on in Huntington's disease. But when you look for this information in HD, it's not much there apart from the cognitive things that have been described.
Because it was mentioned about other parts of the body.
Yeah. It's there. And what's kind of shocking to me is that there's an abundance of this information and excitement about it in Parkinson's disease, and not so much in HD.
We're starting.
Starting, yes. Yeah, but it's not, it doesn't really. Yeah. It's really work that needs to be done. I think we could do more of that and enroll HD, actually, to look for more of that stuff.
The other, just to comment, because Neal was part of this with Huntington Study Group, is this new score, right, that we're going to be using for the clinical trials for this, where they are incorporating some of the cognitive tests, right, and have a composite score. So that might help. I don't know what you think.
Yeah, I think that makes it more. I think this early stuff is still very full exploration.
No, we're still working on filing the IND part with the FDA. So once that happens, which if we get funded to do this, it'll be 18 months from then to start.
Any last questions? I'm sure that Dr. Hermanowicz and Dr. Thompson will lurk around for a few moments.
Thank you. Lurk. I'm sure they'll lurk around for a few moments. But just in case there's anyone thinking I need to get home to something, I think we'll try and wrap it up tonight and just give a last round of applause and thank all of you for coming this evening.